Advanced Higher Biology, Unit 1, Cells and Proteins, specifically Key Area 1, Laboratory Techniques for Biology, covering sections D and E. So detecting proteins using antibodies, immunoassay techniques, what type of antibodies are used for this and why they're useful. We'll look at the actual process of immunoassay techniques, Western blotting, and then we'll finish off looking at two of the types of microscopy, bright field and fluorescent. We should remember from National 5 and higher that the immune system has special cells which produce proteins called antibodies. These antibodies can bind to one specific shape of protein. So each different antibody is specific to one type of protein. Scientists can use these antibodies to produce what are called immunoassay techniques and detect and identify specific proteins. These techniques use stocks of antibodies with the same specificity, known as monoclonal antibodies. A stock of monoclonal antibodies will bind to only the same one type of protein as each other. So you have a consistent and reliable supply of these antibodies. The production of monoclonal antibodies is no longer part of the advanced higher course. However, for interest, it involves combining a certain type of cancerous cell with white blood cells and to make a never-ending supply of identical antibodies. In immunoassay techniques, an antibody specific to the type of protein antigen we're looking for is linked to a chemical label. This label is often a reporter enzyme, which will produce a color change when bound to, but chemiluminescence and fluorescence and other types of reporters can be used as well. In some cases, the assay will use a specific antigen to find the presence of an antibody instead. In Western blotting, we can use this technique after SDS page. The separated proteins from the gel are then transferred onto a solid medium. The proteins can then be identified using specific antibodies that have a reporter enzyme attached. So we can use these antibody techniques to identify different things that we've separated on the gels. Moving then on to microscopy. So bright field microscopy is commonly used, we use it in schools quite a lot, to observe whole organisms, parts of organisms, thin sections of dissected tissues, or even individual cells. And as you can see on the left, it's just a normal microscope which uses light as its method. There's then fluorescence microscopy. So if we compare the microscope on the left here with the one at the bottom right, we can see it's got things like an excitation filter, UV lights, optical fibers, mercury arch lamps, and dichromatic mirrors. So all this together gives us a much, much better view and nicer picture. So it uses fluorescent labels to bind to and visualize certain molecules or cells within structures within cells or tissues. So that's all that's in this section. So we've looked at immunoassay techniques and the fact that they're used to identify specific proteins. These techniques use a stock of antibodies with the same specificity, which are known as monoclonal antibodies. An antibody specific to the protein antigen can be linked to a chemical label. The label is often a reporter enzyme, which produces color change, but we could also have chemiluminescence, fluorescence, and other reporters. In some cases, the assay uses a specific antigen to detect the presence of an antibody. Western blotting is a technique used after SDS page electrophoresis. The separated proteins from the gel are transferred or blotted onto a solid medium. The proteins can then be identified using antibodies that have reporter enzymes attached. When we look at microscopy, we have bright field microscopy, which is commonly used to observe whole organisms, parts of organisms, thin sections of dissected tissues, or individual cells. In fluorescence microscopy, we use specific fluorescent labels to bind to and visualize certain molecules or structures within the cell or tissue.